Welcome to the program. I'm your host, Neil Howard, here on Health Professional Radio. Our guest is Mr. Steve Parkinson. He's president and CEO of Lakewood-Amedex, a privately held clinical stage pharmaceutical company. And he's going to talk with us this morning about their broad portfolio of first-in-class antimicrobial and antifungal therapeutics. Welcome to the program, Steve. How are you?
I'm very well. Thank you, Neil, and thanks for having me.
Well, tell me a little bit about yourself and about the work that Lakewood-Amedex. What are they involved in, and what is your role there, other than CEO and president?
Well, I am a scientist originally. I'm actually a microbiologist, and so I've been involved with the whole world of bacteria for a very long time. Mm-hmm. I started out as a bench scientist, actually, and then got into manufacturing management, business development, and then for the last 22 years, I've been CEO of 4 companies, 12 years with Lakewood-Amedex. The company is, as you know, Neil, a drug discovery and development company. We are discovering new compounds to treat serious infectious disease, bacterial and fungal disease, and we have completed a first clinical trial, and now we're launching into additional clinical trials. We have a new approach to the antimicrobial problem, and as you know, it's the antibiotic resistance crisis has become quite a major issue, and we have a very, very different approach.
So what type of approach does Lakewood take to this antimicrobial resistance? I mean, antibiotic resistance, we've been hearing about it for a long time. As you say, it is a crisis. What is Lakewood-Amedex doing?
Well, the problem is, Neil, with antibiotic resistance is if you look at, how antibiotics all came about, penicillin was the first one discovered in 1928 by Alexander Fleming. But he found it in nature. It was an accidental discovery. And what happened is he found that a mold growing on a plate with bacteria growing on it had a zone of inhibition around it. In other words, a ring where the bacteria wouldn't grow. The fungus was secreting penicillin, which was actually stopping the bacteria from growing. Now, this is nature's way of, or fungus in nature's way of taking care of itself. Bacteria can quickly and easily outgrow fungus. So the fungus produces a toxic compound, penicillin, to stop the bacteria growing. As you know, the problem is we've seen the bacteria not just lie down and take this, they develop a means of resistance. So they can actually destroy the penicillin or pump it out of the cell so that they can continue to live and thrive. And this has happened, unfortunately, with all the antibiotics that we've developed. We have a compound that was also an accidental discovery, but it actually kills bacteria very rapidly. I can tell you a little bit more about it if you would like.
Absolutely.
So our founding scientist, Dr. Roderick Dale, when he was trying to use a technique called gene silencing, it's a way of actually shutting down genes inside an organism to stop them producing things like proteins. So with the MRSA bacteria, which I think everybody has heard of, which is a nasty pathogen, he was trying to inhibit the growth of MRSA by using a gene silencing technique. But the compounds, which were his own compounds that he actually used, he found that they were killing these bacteria extremely rapidly. And what was actually happening is these compounds that we now call bisphosphocins were not actually getting into the cell and inhibiting the bacteria at all. They were actually killing the bacteria very quickly when they contacted the bacterial cell membranes, which is kind of the equivalent of our skin. We found that the membranes were essentially falling apart, rupturing and falling apart, and of course the bacteria died very quickly. Now, the technical explanation, Neil, is that bacteria have a negatively charged phospholipid membrane structure. It's actually somewhat similar to our own, but different enough. The negatively charged phospholipid membrane structure, we actually have a neutral membrane structure. So our compounds are causing a chemical and electrical interference with the structure of bacterial membranes and causing them to fall apart and rupture. So this is a very rapid killing activity, which is quite different from antibiotics. We find, and this has been scientifically, we can kill 10 to the log 6 to 10 to the log 9 bacteria inside of 2 minutes. Antibiotics typically act over a period of a couple of days, 2 to 3 days, and they you have to take them systemically, they circulate in your blood, and then they get to the action site and accumulate back then. So it's a much more rapid, direct approach. There's two things, actually. One is the rapidity of the killing. So bacteria evolve very quickly, particularly compared to ourselves. They divide every 20 to 45 minutes, depending on the species, and they can evolve very quickly. But when you look at the kill times, it's so rapid, they're not even going through an evolution. The other advantage we have is that the core of our molecule, which is not actually the active part of it, is a nucleotide. And nucleotides are essential for life in all organisms, including bacteria. So it would be like poisoning a food that they desperately need, and they can't sort of live without this. So they'd have to go through a massive evolution unknown on this planet to not need nucleotides for growth. So I think we've got them pretty well twice over there.
Does this kill bacteria specifically, or is there ever a possibility of them attacking other cells that have nothing to do with the malady that you're combating?
So they're actually active against bacteria, yeast, and fungi, but there's no activity at all against mammalian cells, our own cells. We've actually proven that quite extensively. This is because of the membrane structure being different. There's two structures. They're known as prokaryotic and eukaryotic. Our mammalian cells are eukaryotic and of a different structure and electrical charge, whereas the bacteria are these prokaryotic cell membranes.
Well, what type of timeline, wrapping up, do you see where these new drugs or treatments are going to be available for widespread use?
So we've completed a first clinical trial on our initial indication, which was a small trial, but actually very, very positive. We saw extremely encouraging trends. We're now pursuing 4 different indications, all localized treatments, and the idea is, of course, that we can prevent a bacterial infection becoming a serious systemic infection by treating the infection locally. And these are going into what we call robust, statistically relevant clinical trials, which means we get a very, very good indication of their efficacy and the fact that they work. And we should see results from those by mid-2020. So we will know that we've got very effective compounds by then. And then we should see drugs coming to approval around 2023, which sounds like a long time away, but it's actually not. In drug development, that's a very short time.
Well, where can we go online and learn some more?
We have a website, which is www.Lakewood-Amedex.com, and I'll say Amedex is AMEDEX. And you can get a lot more information there.
Steve, it's been a pleasure. Thank you for joining us on the program today.
Thank you, Neil.
You've been listening to Health Professional Radio. I'm your host, Neil Howard. Transcripts and audio of this program are available at hpr.fm and healthprofessionalradio.com.au.
Thank you for listening to Health Professional Radio. We're very proud to be an independent broadcaster providing our content free of charge to you, the listener. One of the ways that we're able to remain free and independent is by having people like you become patrons. You can support Health Professional Radio simply by visiting hpr.fm and clicking the button that says Become a Patron. Your patronage of even just $1 a month lets us know that you're there, which in turn makes us more valuable to advertisers. And, of course, if you're able to afford more, then we would certainly appreciate the support. My name is Toby Longhurst from Health Professional Radio. Please visit hpr.fm, click the Become a Patron button, and support us if you can.